All right, so our next speaker is Dr. Kara Margulis, and she is somewhat of a rarity in pediatric GI, in that she's also a very accomplished basic scientist, something we definitely need more of. She's an international expert in how neurotransmitters affect the GI tract, and she focuses on their role in children with autism. She's currently a professor at Columbia University, and we're very happy to have her here and to tell us about the much more scientific part of these disorders. Welcome, Dr. Margulis.
Thank you, Bill. I'm just going to share my screen. Okay. So I first off wanted to thank the organizers for letting me be a part of this. It's been, a real pleasure and honor, not only to hear everyone else's talks, but really to hear the family perspective too, which I think is so important, as a physician and as a parent. So thank you for letting me be a part of this. And I'm sort of happy to give the last talk, which is unusual because I feel like between the 3 talks that came before me, that it sort of gave a lot of background to what I'm going to go into today, which is the brain-gut microbiome access in autism spectrum disorders. So my disclosures are that I consult for Scioto and Biomica, and I serve on several editorial boards, but those are not reflected in the talk I'm going to give today. And as Bill said, I'm a pediatric gastroenterologist with a clinical interest in gut-brain axis disorders, and really chief among those is GI problems in children with autism. And I've been really fortunate to be able to segue my clinical interests into a research focus, looking at this relationship between gut issues, the brain and behavior in autism, with a more recent focus on the microbiome brain-gut axis, and I'll talk a little bit about that today. So I just wanted to start with a quick patient presentation of a child I now saw quite a few years ago. He was a 17-year-old with autism who came to me with a history of recent regression. So 6 months prior to his visit, he was verbal and was really a genius, both in math and reading. He went on a family trip to India where he caught a gastrointestinal bug, which presented as diarrhea for 5 to 7 days. And while the diarrhea resolved with antibiotics, his behaviors radically changed. He became completely nonverbal and stopped the reading and math, which he loves so much. He also developed these severe obsessive compulsive behaviors, one of which was compulsive water drinking. And he was actually admitted to the intensive care unit several times because of water intoxication. But upon history and physical, I noted that he had a variety of signs and symptoms that are related to what we see sometimes in kids with autism and GI distress. He banged on his chest, he had excessive drooling, and he had really difficult stooling with self-disimpaction. So I treated him for severe constipation and gastritis, and all of these relatively acute onset behaviors resolved once his GI conditions were treated, including that obsessive water drinking and also that catatonic-like behavior. So I'm gonna, what Kent and Andre talked about at the beginning was that individuals with autism more commonly are diagnosed with GI conditions. The clinical presentations of GI distress can be dramatically different than kids that are typically developing. And it's important to understand these distinctions, not only for diagnosis, but also treatment, which I'm going to focus on today, because treatment options, as we know, are highly limited in many. And even if we can treat the GI condition successfully, a lot of times challenging behaviors persist. And we've seen increasingly that gut problems are linked to behaviors, mood, and cognition. So, as Ken said, a lot of these kids, we think, as opposed to having a brain disorder, have a brain-gut disorder, and treatments need to consider both, and gut-brain treatments are highly limited. So what I'm going to talk about today is really just quickly review the gut-brain axis and then the microbiome's role, addressing the question, is there a specific autism microbiome, and can we fix the gut microbiome in autism? And then go a little bit into nutrition in autism. How is nutrition linked to the gut microbiome? How can diet in autism be studied in a way that reflects the diversity of individuals with ASD? And finally, a little bit on our research of whether a nutritional intervention may actually be able to change the GI and behavioral outcomes in children with autism. So the gut-brain axis, which Ken and Andre both referred to in their talks, really is this continuous bidirectional communication, which links gastrointestinal symptoms in autism to the behavioral symptoms. And there's been a lot of work done on the gut-brain axis, but three major mechanisms have been studied more than all the others, and that includes serotonin, the immune system, and bacterial molecules, or the gut bacteria. And like I said, I'm going to be focusing on bacterial molecules today, not only how it relates to nutrition, but bacterial molecules have a very strong interplay with gut neurotransmitters and modulators. And so peripheral serotonin is one of them and is really another expertise in my laboratory. So what is the gut microbiome? On the simplest level, it's basically the ensemble of bugs in your gut. And so the gut microbiome is huge. There's over 100 trillion microscopic life forms living in your gut. And so it's not only huge, but it's incredibly complex and individualized. The microbiome's as personalized as a fingerprint. So no one person has a microbiome that's exactly like another, or at least that we're aware of. And this is a phylogenetic tree. So what it does is it looks at all the different types of all the different autism studies over a 10 to 20-year period, which look for microbiome differences in individuals with autism compared to those without. And what you can see here is that there are differences in many of these studies, but what you also see is that the differences are very different from one another. Once again, really emphasizing this fact that there's no one unique autism gut microbiome. So how do we fix the gut microbiota? And this slide shows a lot of the different attempts that have been tried. And, probiotics is one of them, where you give one bacteria or a group of several bacteria to the gut. But a lot of clinical questions remain. So which gut bacteria are the one that actually work? How much do you give? And there's 1 trillion bacteria there. So what are your targets? Different bacteria, as we're seeing through research, really has different effects, both in the gut and the brain. So figuring out which one affects which systems is still a challenge. Similar with the microbial metabolites, so what the kinds of sort of chemicals or neurotransmitters that probiotics or any sort of bacteria give off, the same clinical questions arise. Which one, how much, and again, which ones target problematic behaviors or could be potentially be dangerous or not helpful. So another approach has been, let's give all the bacteria through fecal microbiota transplantation, and that's really giving stool from a quote healthy individual to somebody that has a medical issue. And so one study has been done in this in kids with autism. It was a small study. It had very exciting findings that it did seem to improve even long-term gut and behavioral outcomes. But that study needs to be optimized. We need a larger study, something where there's a placebo control, because we know in prior studies where we look at autism that placebo does make an effect, and other issues like that. There's also transfer of unknown bacteria. And so there's been a number of studies now which have started in sort of the preclinical or animal study realm, but even in humans now, that when you transfer unknown bacteria, you might not only have good effects, but you could have negative effects. And some of those that have been shown include mood, obesity, infection, and more recently, even potentially autoimmune disease. So finally, we go to diet, and there's been a lot of talk of which, or alluding to nutrition, and Kay certainly talked extensively about that in Rett syndrome in the talk right before mine. And the reason diet's attractive is because it's very challenging to feed a lot of children with autism, but what factors are modifiable that we can really use to change that gut microbiota? And what you can see from the figure here is that most of these are not modifiable. It's hard to live in a specific geographic location for this reason. You can't alter host genetics or your aging, or even the way that you were delivered as a baby. But there's a couple here that can be attached, like exercise, stress, but diet is something that's readily approachable. And as Andre alluded to in his talk earlier on, autism there is highly complex, and really no one child or adult with autism is like another. And so really, when you have an autism-based study, you really need to consider that diversity in autism. And this was one study published a couple of months ago in Cell, which really looked at this and finally considered all these different variables. So they looked at the clinical backgrounds of these patients, what medications they were on. They looked at nutrition to some degree, and then they looked at the stool microbiota. So again, their study confirmed this finding that there's no one unique autism-associated microbiome. But what they did see was an effect on diet. So they saw that when individuals had a less diverse diet, so that means less variety in the diet, that led to a less diverse microbiome, and that led to looser stool consistency. That was measured by one bowel movement taken. And so really what this showed is that autism-related dietary preferences, or really food variety, mediate autism gut microbiome associations. So this was definitely a big step forward in being able to sort of say or recognize that autism is complex. But there are a lot of questions that this study didn't address. There was really no dietary detail with regards to gastrointestinal or behavioral outcomes. And diverse is a very general term. It doesn't answer the question, which foods or food groups should you ingest or avoid? And it was also one stool sample, which as a gastroenterologist, we know can vary between diarrhea, constipation, and as Ken talked about, diarrhea can sometimes present as, constipation can sometimes present as diarrhea. So you really need to get a better look at that. And so it's important to do this too, because as you see here, all these different components of diet have very different effects on the microbiota. And this really needs to be looked at more in depth in children with neurodevelopmental disorders. So a study that we have been intimately involved in and that was funded by Autism Speaks is beginning to really look at all this. And, I'm going to, before I introduce the study, say that I'm going to give general big picture findings because we're writing it up right now and I can't talk about the details, but I want to give hope to families and say that I think the studies are out there. It's a multi-center study. Ken Williams was the principal investigator at Nationwide Children's. Tor Savage, who's by Texas Children's and who Dr. Modal knows well as Ruth Ann Luna, are there. And at UT Southwestern, Dr. Powell was there, and I've been the principal investigator at a residential center for kids with autism called the Center for Discovery. And I put Ruth Ann Luna's picture up in the upper right-hand corner because she is the principal investigator in this study. So almost 250 children were recruited. About 50% have autism. The remainder are neurotypical. And we did a comprehensive evaluation of all these kids. So comprehensive behavioral assessments through 4 different studies, in comprehensive gastrointestinal assessment, food diaries for 2 weeks, and looked at all those different active medical issues and medications. Took a stool specimen and then looked at the microbiome. And again, that metabolome, so those chemicals or neurotransmitters that the microbiome can affect or be affected by. And then did something called a multi-omic analysis, which is allowing us to look at how specific parts or which specific bugs in the gut microbiome or which specific mediators in the metabolome correlate with gastrointestinal problems, behaviors, and other medical issues, and nutrition. And while I have nutrition listed last there today, I'm really going to be focusing on nutrition. First, as a result of the study of the 3 outpatient populations that we looked at, and then looking at a dietary intervention with the Center for Discovery. So what was seen in these kids from outside, again, the Center for Discovery, was that when you look at food diversity or a food variety score, what you see is that these kids with autism that you have highlighted in that bluish purple color have a lower food diversity compared to both their siblings and typically developing kids who are not their siblings, but it was significantly different in those kids who were typically developing and unrelated. And unlike the paper in Cell, we saw no significant microbial associations with stool consistency, likely because of the things Ken talked about and the idea that we looked over a longer period. However, importantly, we saw microbiota differences or food variety differences associated with other GI-associated symptoms, including increased severity. And we also saw in this large population increases in behavioral challenges with GI symptoms across all groups. So then we looked at these microbiota differences and look for correlations with specific dietary components. And ASV is just a fancy name for a state-of-the-art way of really being able to detect the different microbiota. And what we saw was that there were several ASVs that were associated with these increased food variety scores. And 28 of them correlated with 24 different behavioral variables. So you can see here some of the behavioral variables we see, including lethargy, touch sensitivity, sensory seeking, compulsive behavior, and irritability, really showing that different microbiota may modulate different types of autism-associated behaviors. And similarly, we saw others that correlated with core diagnostic features of autism, including, I have listed here, stereotypy, really showing that this supports a personalized approach to really how did you research with these individuals and also potentially for treatments. So to determine whether nutritional intervention plays a role, we again looked at this residential autism population with an in-depth dietary intervention. We compared them to age and sex-matched subsets, both in the outpatient autism population as well as the typically developing kids. And then what we found was significant improvements in gastrointestinal symptoms and behaviors observed in this residential cohort compared to the other cohorts of kids with autism. And they were significantly associated with differences in both the microbiome and the metabolome. So first, what you can see is that, again, in that bluish purple, you have the kids with autism who are on the outside of the Center for Discovery. And in red, you have the kids within. And what you can see is that their food variety scores are much increased compared to the kids with autism, and actually, and no different from the typically developing kids. We saw microbiome differences at multiple levels. I'm showing here the phylum and the genus taxa levels, and we saw that they were related to not only food diversity, but also specific food groups, both when we compared the kids with autism in and outside of the Center for Discovery, and also in the kids with autism versus those that are typically developing. And then we saw differences in metabolites. So this is something called a principal component analysis. And each shape there represents an individual, and where they're located on the figure represents their entire gut microbiota and sort of where they're related. And what you can see is that the kids at the Center for Discovery have a very different metabolic profile compared to those kids outside the Center for Discovery with autism and those that were typically developing. However, there were limitations to this, and that there was really, this is called a cross-sectional study. So it was really a snapshot at one time for all of these individuals. And that leaves us with limitations. Do the behavior and GI outcomes influence the gut microbiome or the metabolites? Or on the opposite end, do changes in the gut microbiome and metabolome from, say, diet affect behavior and gastrointestinal outcomes? So what's needed is a prospective longitudinal study, and we are doing a prospective study like this right now, but of course it's in the very preliminary stages. So in conclusion, GI problems, as everybody on this call knows, are common in autism, and that treatments for GI issues in individuals with autism can be complex because autism itself is highly complex, but brain-gut interactions are likely involved in many cases. Gut microbiota may be a target for both GI and behavior issues in autism, and maybe even the core features of autism. There is no autism microbiome that's been detected thus far, but personalized approaches in research and treatment, I think, are going to be key based on this study and that other one that I presented here. And families need more specific guidelines and more targeted approaches. So interventions that can be implemented in line with the challenges of autism certainly are warranted. And nutrition may be an option as we begin to cipher out exactly what we need to give. And more specific nutritional interventions may change the course of autism, not only the core traits, but also behavior and gastrointestinal outcomes. And again, types of foods may make a difference. And like I said, I'm highly optimistic. There's more to come, hopefully within the next few months after we submit this paper as well. I just want to thank the team at the Center for Discovery that really has helped lead the clinical studies from my side. And of course, I've highlighted in yellow here the key collaborators on this study. And of course, my major collaborators in other areas, science is done in a group, which is why I appreciate a meeting like this, which brings together people from different backgrounds. And of course, I can't do anything without my research support. And so now that we're done, I'll take any questions. Thank you.